Knowing how to read a cladogram is half the battle. This is video 3.2 about making cladograms. We'll cover these three topics. Hit pause now for more details. Cladograms are diagrams that show evolutionary relatedness, but the sources of information used to make cladograms can vary. Now, cladograms will mostly be made on shared morphology, similar structures, and molecular similarities like proteins, DNA, and RNA, but still, even more information about fossil ages, biogeography, and behaviors can be used to make cladograms. This is a trait table. The different species or taxa are on top and the traits are on the side. Now, in a trait table, a 0 or a minus means an organism doesn't have the trait, and a 1 or a plus means the organism does have the trait. When we talk about these traits from an evolutionary sense, we call them derived traits. A derived trait is one that arose during evolution and is being used to separate organisms into different clades. It's important to clarify a rule here. The more derived traits two species share, the more related those species are. And this is important when setting up cladograms. Another key understanding is the concept of an outgroup. The outgroup is the species with the fewest derived traits shared with the group. Now, the outgroup is a reference point because the outgroup is a lineage just outside of the other organisms being considered in the analysis. Now let's make a cladogram. It's okay to reorganize or write on the trait table as you go. And let's start by drawing this cladogram V shape, and then find the outgroup. The outgroup is lancelets, which shares no derived traits, so write lancelet. Then write the derived trait that comes after the lancelet. Everything else has a vertebral column. Then write the derived trait that comes after the lancelet. Everything else has a vertebral column. Cross that info off just to get it out of the way, and then it's like finding the outgroup of what's left. Lampreys only have vertebral columns, but no other derived traits. So lampreys go on the next tip, and everything else has the derived trait of jaws, so get jaws up there too. Now cross off lampreys and jaws, and the next kind of outlier of what's left is the tuna. Tuna are the only ones left that don't have 4 legs. Get that up there, cross out tuna, cross out 4 legs, and do you see a pattern here? Next are salamanders, which have these 3 derived traits, but not amniotic eggs. Boom, now we've got turtles who have all of these traits, but not hair. And then we can finish off this trait table by adding leopards on the last tip. Our cladogram is complete. Notice that each tip has a species on it here. Trait tables are rarely that easy, but the basic idea for making cladograms from trait tables is the same. Check this one out. Right away, I noticed that lampreys are the outgroup with all of these minuses, so let's start there. Here we go. Now, all the other species have jaws, so get jaws on that cladogram, and then let's cross out what we've used. So jaws, and then right on the cladogram, now pike sticks out as kind of like the next outlier. Pike is the only species without lungs. So get pike and lungs on that cladogram and cross out that data to make things easier to see. Now lungfish is really sticking out. But wait, what does everything else have that lungfish don't? Lungfish and turtles don't have hair. Lungfish, gorillas, and wolves and humans all don't have shells. And lungfish, wolves, and turtles all don't have grasping hands. So in cases like this, you'll have to divide your remaining species into 2 groups. Check it out. Okay, so put lungfish over here, and now almost everything else has hair, so I'm gonna put hair over here. But turtles don't have hair, so where do turtles go? No problem, just have turtles as a branch off the lungfish. Like this, but remember, turtles have the trait of bony shells, so we gotta get that on there. Okay, I know it looks weird, but it's a legal move, and you'll get used to it. So let's cross off all of that info. Lungfish, turtles, bony shells, and hair. All right, now regroup, and you'll see that next are wolves who have jaws and lungs and hair, but no grasping hands. Get that on there and cross that information out, and we're left with gorillas and humans. Now, both have all 4 of these traits, but there's no unique traits to distinguish them. No problem, just put each on a separate tip, and it doesn't matter which side you put them. Let me show you. All right, so I put humans on the left and gorillas on the right, but you could have just as easily flipped them and the cladogram would mean the same thing. Most cladograms nowadays are made by comparing molecular sequences. Now, this could be DNA or protein or RNA. Think of sequences as kind of a molecular clock. Remember, DNA accumulates random mutations. So as two species are separated, each species accumulates different random mutations. And over time, these differences accumulate. Some evidence suggests that the rate of accumulation is fairly steady, so the number of differences can be used to determine how long ago speciation events were. But all right, we're getting away from the point. The simple rule here is that more molecular similarity means more closely related, and more molecular difference means less related. Let's use that rule to make some cladograms. Here's some sequence data. We're comparing different species that are represented as letters, A through D over here, and V through Z over here. The table on the left shows percent DNA homology, so percent similarity, and on the right is number of DNA differences. So species A and species B have 68% DNA similarity, A and C have 42% similarity, and so on. So how do we turn this into a cladogram? First, figure out if you're dealing with similarities or differences, and next, apply the right rule. For similarities, high numbers mean more related, and for differences, high numbers mean less related. And use these rules to identify the outgroup and also identify closely related species. Let's try each. So this table shows similarities. With similarities, high numbers mean more related. So the outgroup then has the lowest similarity numbers compared to all species, and that's C. At most, C has 42% similarity with another species. So C is the outgroup. We'll put that there, and now find the most closely related with the highest DNA similarity. So A and D. Now we're ready to start the cladogram. Basic cladogram shape, let's put C as the outgroup. There we go, and A and D are most closely related. Right on there, and B is somewhere in the middle. So this cladogram summarizes this information. With some practice, you'll be a pro. Let's check out the other scenario. Similarities or differences? Well, this is DNA differences. So high differences means less related, and low differences means more related. The outgroup would have the highest differences compared to other species, so it looks like that's species W here that has all the highest numbers. Write that down here, and now closely related species have the lowest numbers. Right here, X and Z only have one difference, and V and Y only have two differences. So those two sets are closely related. And now that we're organized, onto the cladogram. Basic starter cladogram, let's get W on as the outgroup. Next, get X and Z on there as the most closely related. Here we go. Now remember, I could have put X then Z or Z then X, and it means the same. Now, we have to show that V and Y are also closely related. Well, just put V and Y close to each other too. There we go. Now we've got a cladogram that tells the story of our molecular data. So let's recap. Most cladograms are made from molecular or trait comparisons. Now, with traits and molecules, more similarity means more relatedness, and also more differences means less relatedness. Finally, molecular changes can be used like a clock, where the number of differences can actually be used to estimate the time since speciation occurred.